this video, we'll study about the structure of the antibody. Competency is again the same. So what's the structure? Antibodies are Y-shaped heterodimers and they are composed of 4 polypeptide chains. I will show you the figure here. You can see there are four polypeptide chains, and these are some of them are small and some are huge. The small one is called as light chain and the huge one is called as heavy chain. So we have two heavy chains and then two light chains, and these two are connected by disulfide bond. So actually there are two different types of disulfide bond, and one is called as inter chain disulfide bond, and again another one is intra-chain disulfide bond. Interchain disulfide bond connects two different types of chains, that is one heavy chain and the one light chain, and uh, two different two different heavy chains at the hinge region, but the intra-chain disulfide bond connects within the chains, so they will connect within the chain, the same chain, either heavy or the light chain. And these heavy chains and the light chains are divided into different types. The heavy chains are alpha, nu, gamma, delta and the epsilon. Based on these heavy chains, immunoglobulins are classified into different types, and the name of the each immunoglobulin types is given based on the first letter of the uh, heavy chain type. So you can see IgA alpha. The first alphabet is alpha, sorry A, and then the light chains are divided into two different types, kappa and the lambda. Okay, and we need to know that each heavy chain and light chain has two different regions. Those are called as variable and the constant region. If you observe this figure, you can see the pink colored region and then the blue colored dark blue colored region. So these are the two different variable and the constant portions. Okay, so what is variable? Here amino acids are variable as the name suggests, and this is present near at the end terminal. So this is our end terminal and this is carboxy terminal. So this is the N terminal and this is carboxy terminal. The constant region is having the constant amino acids, while variable region has variable amino acids. As you know, this variable region binds to the antigens. The antigens are of different types. So to fit to the antigen, we need different types of amino acids. The same type of amino acids won't work. And then we call them with different names, VH and VL for variable heavy chain and variable light chain, CH and CL for constant heavy chain and constant light chains. And we will move to the domains now. So domains, we have heavy chain domains and light chain domains. These domains are nothing but further folding of the chains itself. Okay, and we have a variable light chain domain and constant light chain domain. In the same way, variable heavy chain domain and three constant heavy chain domains. It's not true for all heavy chains, because as you can see here, the nu and epsilon have four constant domains. So these are the four constant domains for nu and epsilon heavy chains. Let's talk about the hinge region. Whatever the object may be, these hinges allows the object to work properly by giving it a rotation, flexibility, twist or anything. In the same way, this hinge region is quite flexible and allows the different position for Ig molecules to bind with the antigens. So these hinges are helpful. But we need to notice that nu and epsilon heavy chain do not have hinge region. Instead of they have one extra domain. So enzymatic digestion. We have three different type of digestion. As you know, these antibody is nothing but the peptide chain. So it can be digested by various enzymes. So we have peptide digestion, pepsin digestion, and the mercaptoethanol digestion. So what this does? As you know, this peptide digestion, it involves the cleavage between just above the hinge region where disulfide bridges are below it and it's not involved. So once if that cuts just above the hinge region and above the disulfide bridges, these two antigen binding fragments will be separated. So you can see AB refers to the antigen binding, and the below one is partially digested, and this fragment is called as FC because it crystallizes in the cold and it's insoluble, but the two FAB fragments are soluble. Now looking into the pepsin digestion. This pepsin does actually opposite to the papain. Actually what I said was papain cuts above, but this pepsin is going to cut below the hinge region. One hinge region below the disulfide bridges. Once if you get into that imagination, as you can think, if I cut here, these two fragments will be together. So we will get two FAB fragments combined together as one. So one FAB2 fragment composed of two FAB units bound together, and so many smaller fragments due to the digestion of FC portion by pepsin into the smaller fragments. And we left with mercaptoethanol. Here what it actually does is that it breaks all disulfide bonds. Once if it breaks all disulfide bonds, what are the function of disulfide bond? Actually they connect two different peptide chains. So we are getting the peptide chains are getting separated. Now we will get four different chains. Two will be light chains and two will be heavy chains. Okay. Then what are the functions of the antibodies? These are just one line. You can paste it anywhere in near wall or mirror, anywhere you want. Just write these down. These are directly from the GK. Okay, functions of the antibodies. The first one, IgG. It's the complement fixation by classical pathway. As you know, what is you don't know what complement is. Just I am going to make a video on that later. Just wait for that. And IgA is a localized protection in the body for by secretions like tear, salivary secretion, intestinal secretions, etc. But mainly if you heard the word IgA, just remember secretions. And it also functions by complement fixation by alternate pathway. So we have different pathways in complement fixation that we will learn soon. And IgM, it works by complement fixation by classical pathway, and IgD is present on the B cells, so help in the antigen recognition. And IgE, it releases histamine from mast cells and the basophils. It also has reagin activity, nothing but the substance which is involved in uh, allergic reactions. That's it. This is the end of the video. Thanks for watching video completely, and if you have any doubt, feel free to ask in a comment box or you can mail me. Thank you.